And as you can see from the arrows that these two are isomers of each other, right? With this isomer glyceraldehyde 3-phosphate being more stable, so ultimately what happens is this dihydroxyacetone phosphate also changes into glyceraldehyde 3-phosphate. This phosphate will be given to this ADP, this phosphate will be given to this ADP, these two ADPs will become ATPs, so we will get 2 ATPs in this step as well. In this process, I will discuss each and every step with proper visualization of what exactly is happening there. So pay very close attention, right? And everything will be clear after watching this video. Hello everyone, my name is Shivansh Gaur and welcome to ATP STEM. In this video, we are going to discuss glycolysis. In my previous video, I discussed aerobic respiration versus anaerobic respiration. If you have not watched that video yet, I would suggest you to go and watch that video first. I will be giving the link of this video in the description below, right? You can click on the link and watch that video first, and then this video will make much more sense to you. Okay, so now let's start that what is glycolysis. Like I told you in my previous video that glycolysis takes place in the cytoplasm. It does not require oxygen and it is common pathway for both aerobic and anaerobic respiration, right? So term glycolysis, like where has this term come from? So term glycolysis has originated from the Greek words glykos, which means sugar, right? And lysis, which means splitting. So which sugar we are talking about? We are talking about glucose, right? Here, one molecule of glucose breaks down into 2 molecules of pyruvic acid, okay? So therefore the term glycolysis. Also called as EMP pathway because it was given by 3 scientists named Gustav Embden, Otto Meyerhof and J. Pernas. So from Embden, Meyerhof and Pernas, we have taken EMP. So therefore, it's also called as EMP pathway. Like I told you that in this process, one molecule of glucose will be broken down into 2 molecules of pyruvic acid in the absence of oxygen, as in oxygen is not required in the cytoplasm. Also, there will be net gain of 2 ATP here in this process. Okay, so you can see glucose, which is a 6-carbon compound, in the cytoplasm, it will break down into 2 molecules of pyruvic acid and 2 molecules of ATP will be given. And this 2 ATP is basically net gain. Why it's called as net gain? We will talk about it. So now let's start with all the steps of glycolysis. It is a 10-step process. In this process, I will discuss each and every step with proper visualization of what exactly is happening there. So pay very close attention, right? And everything will be clear after watching this video. Okay, see. The first step in the glycolysis is when glucose, which is a 6-carbon compound, now here you see I have made these ball-like structures. These ball-like structures are basically representing the number of carbon atoms. Okay, so you can count 1, 2, 3, 4, 5, 6. There are 6 carbon atoms in the glucose. Okay, now in this glucose, one phosphate is added. And where does this phosphate come from? ATP. ATP is adenosine triphosphate. It has 3 phosphates. Okay, it gives its 1 phosphate to what? To glucose. And where does this 1 phosphate attach to? The 6th carbon, right? This reaction is mediated by an enzyme called as hexokinase. So finally, we get glucose 6-phosphate. After this step, you can see that there is a phosphate attached to the 6th carbon atom here. This reaction is mediated by hexokinase. Please do not forget that. Now what happens next? We got from glucose to glucose 6-phosphate. Now what next? So basically, just isomerization of glucose takes place. Glucose, which is an aldohexose, it gets transformed into fructose 6-phosphate, which is the ketohexose. So it transforms into, or changes into its ketose form. Okay, so we get fructose 6-phosphate. So basically it's an isomer, so the total number of carbon atoms will remain the same, 6 carbons, right? And there will be a phosphate attached to the 6 carbon. So this, after this step, we get fructose 6-phosphate. Okay, now see, very important. This fructose 6-phosphate gets one more phosphate from ATP, right? Will give its one of its phosphate to fructose 6-phosphate, and it will attach to the first carbon of the fructose, and from fructose 6-phosphate we will get fructose 1,6 bisphosphate. See, there is a phosphate on the first carbon, there is a phosphate on the 6th carbon. This is fructose 1,6 bisphosphate. Now you must be wondering that why it's bisphosphate and not biphosphate, because you will come across some terms which says biphosphate, and there are some terms which says bisphosphate. So what happens is, when there is a gap of carbons in between two phosphates, then it is called as bis. But if the two phosphates are attached to the very next carbon, like very next to each other, then it would be called as a biphosphate. Here we call it a bisphosphate. Why? Because there are carbon atoms in between. Okay, now see, fructose 1,6 bisphosphate, which means fructose has two phosphates on the first and the 6th carbon. Now after this reaction is very important, because now the fructose 1,6 bisphosphate will break, right? Okay, into two molecules of triose phosphate. So we get two triose phosphate, we get glyceraldehyde 3-phosphate, and we get dihydroxyacetone phosphate. And as you can see from the arrows that these two are isomers of each other, right? With this isomer glyceraldehyde 3-phosphate being more stable, so ultimately what happens is this dihydroxyacetone phosphate also changes into glyceraldehyde 3-phosphate, which means that after this step you will get two molecules of triose phosphate. Which one? Glyceraldehyde 3-phosphate, because this one will also convert into glyceraldehyde 3-phosphate. This is how it looks. See, 3 carbon atoms and a phosphate here, 3 carbon atoms and a phosphate here. Right? Clear? Now next step becomes super important, because this compound is going to get added with one more phosphate. And you can see here that this triose phosphate, that is glyceraldehyde 3-phosphate, this one, right, it will take two inorganic phosphates from phosphoric acid. It's called as inorganic phosphate. And one phosphate will attach here as well. Okay, along with two molecules of NAD plus gets converted into two molecules of NADH plus H plus. Why 2? Because like I told you that this triose phosphate, that is DHAP, dihydroxyacetone phosphate, will also convert into glyceraldehyde 3-phosphate. After this, see here, therefore we will get now two molecules of triose bisphosphate, because here 1,1-phosphate got attached to both of those glyceraldehyde 3-phosphate. Okay, so now we have a phosphate at the first carbon as well and at the third carbon as well. Therefore bisphosphate, how many molecules? 2 molecules. So these are 2 molecules of bisphosphoglyceric acid. Okay, so now see here, total 2 phosphates on one bisphosphoglyceric acid and 2 phosphates on another bisphosphoglyceric acid. Next step becomes very, very important. Why it becomes very important? One more thing. Did you see something very interesting? See, in the first step and in the third step, ATP was utilized. ATP was utilized. Now you must be wondering that, sir, glycolysis was supposed to give us ATPs. Right? It is supposed to give us 2 ATPs, but it is utilizing, it is taking that ATP, consuming that ATP. So how do we get those ATPs? No, don't worry about that. Why? Because see, after this after this step where we have bisphosphoglyceric acid, what happens is 1,1-phosphate from the first carbon of both the bisphosphoglyceric acid is given to ADP here. See, so ADP here will get a phosphate from the first carbon here, and this ADP will also get the first carbon from here, right? And ADP will get converted into what? ATP, that is adenosine diphosphate will take one more phosphate and become ATP, adenosine triphosphate. Okay, now see, after these phosphates are given to ADP, what did we get? We get 2 molecules of ATP here. Now see here, this triose phosphate it still has 2 phosphates attached to it at the third carbon, so it's called as 3-phosphoglyceric acid. What it's called? 3-phosphoglyceric acid. Right? Now see, the next reaction is basically a mutation reaction. This phosphate moves from the third carbon to the second carbon. Right? That's the only thing which is happening in the next step. Right? So from 3-phosphoglyceric acid, we get 2 molecules of 2-phosphoglycerate. See, 1 and 2. 2 molecules of 2-phosphoglycerate, that is glyceric acid. It's an acid. Okay. Now see, next step is also very important because this glycerate will change into enol pyruvate. What is enol? You must be wondering. So basically, enol is when OH group, that is alcoholic group, is attached to the double bonded carbon atom. It's called as enol. En for double bonds and all for alcohol. Right? Now when you remove this hydrogen from here and attach a phosphate here, it becomes enol pyruvate. Okay? So here we get phosphoenol pyruvate. As you can see here, that the second and third carbon atom have double bonds, and on the second carbon atom, right, we have oxygen, and instead of hydrogen here, right, hydrogen is gone and phosphate is attached. So we get 2 molecules of phosphoenol pyruvate. Okay, right? So these are 2 molecules of phosphoenol pyruvate. They still have 1,1-phosphate left with them. Now what to do with these phosphates? Very simple. Give them to ADP again. So what will happen? See, this phosphate will be given to this ADP. This phosphate will be given to this ADP. These 2 ADPs will become ATPs. So we will get 2 ATPs in this step as well. And these 3, like these 2 molecules of triose phosphate now becomes what? Pyruvic acid. A 3-carbon compound, 2 molecules of pyruvic acid. Right? So see here that we started from 1 molecule of glucose which contained 6 carbon atoms. Then we added some phosphates, right? And when the lysis took place, like when these phosphates got removed, ultimately we got how many ATPs? 4 ATPs, because count, see, we got 2 ATPs here in this step and we got 2 ATPs here in this step. So total how many ATPs? 4 ATPs, right? But we utilized 1 ATP here in the first step and 1 ATP here in the third step, right? This phase, when the ATP is utilized, is called as preparatory phase, right? And this phase, when the ATPs are synthesized here and here, they are called as payoff phase. So it's like first 5 reactions are called as preparatory phase. The next 5 reactions are called as payoff phase, right? So how many ATP we get in payoff? 4. How many utilized in preparatory? 2. So we can say that we have net gain of 2 ATPs, right? Because we got 4 but 2 were consumed. So we have a net gain of 2 ATPs during the glycolysis. So if we will make the glycolysis balance sheet, then for 1 molecule of glucose, we will utilize 2 ATP, right? But 4 ATP will be given during payoff phase, right? And therefore there will be net gain of 2 ATP. Okay? Apart from ATP, one more very important thing we were getting, and which was in this step 2 molecules of NADH. It will be utilized later in electron transport system. I will talk about that in my electron transport system video, right? But for now, you just need to know that apart from ATP, we are also getting NADPH, which is going to play a very crucial and important role in aerobic respiration. Okay, so here we will write that we get 2 NADH as well, which will be used later in ETS, that is electron transport system. I hope this glycolysis was clear to you with this kind of visualization, because generally you are wondering and you are imagining molecules, right? So this way it is better for you to understand that how exactly the phosphate is moving around, right? And what exactly is happening there, right? Now after this you should be ready to do some questions on glycolysis. So let's quickly do some practice question. The first question here is, for 2 molecules of glucose, glycolysis uses and produces how many ATP molecules? Now see here, for how many molecules of glucose? 2 molecules of glucose. Okay, what we just discussed was for 1 molecule of glucose. For 1 molecule of glucose, we just discussed that what happens? 2 are utilized, right? 2 are utilized and 4 are produced. But for 2 molecules, 4 will be utilized and 8 will be produced. Simple, right? Multiplied by 2, right? So here the correct answer for this question would be this one, that is 4 and 8, A option, right? Now let's look at the next question. Next question is, how many molecules of NADH are produced, right, when 4 molecules of phosphoglyceraldehyde are converted into 4 molecules of pyruvate? Now see again what they are asking. They are asking for 4 molecules of pyruvate, and the glycolysis that we discussed was for 2 molecules of pyruvic acid. Okay, so how many NADH we were getting in 2 molecules of pyruvic acid? 2. Remember, 2 NADH we were getting. I just told you here 2 NADH we are getting. Okay, so for 4 molecules of pyruvic acid, simple. How many? 4. So the correct answer here would be 4. Right? We will be getting 4 NADH molecules. Next question, very simple, very simple. The enzyme which converts glucose to glucose 6-phosphate. This was the first step. Remember, I told you that, please remember that this step was mediated by an enzyme called as hexokinase, right? So the correct answer would be here hexokinase. Okay, C. And the last one, very simple question, is what is the other name of glycolysis? I talked about this in the very first part of my video, right? It is EMP pathway on the names of the 3 scientists, Emden, Meyerhoff and Pernas. Okay, so I hope the glycolysis was super clear to you. I will attach the PDF of these notes in my description box, right? So you can download this PDF, right, for your reference whenever you want. My next video will be on Krebs cycle, right? So please subscribe this channel. Like this video, share with your friends, right, whom you think will be of great help when they get this video, right? So share it with your friends. I will meet you in the next video with Krebs cycle. Till then, take care. Bye-bye.